Yes, hopefully my talk will be open. Great. So I'm sorry that I have to wake you up after the dinner, after the lunch without coffee, which is really hard, I believe, at least for me, for sure. So after the cardioprotection in the morning, now we go to cardiotoxicity. And I will give you a very simple presentation, I guess, and on some new paradigm changes in safety testing and what is cardiotoxicity. Basically, this is conflict of interest. I'm also running some small companies as well. Tomorrow I will talk a little bit on it. And basically, it goes. So basically I would like to show you this recent paper that we published with Rainer Schulz's group recently in the European Heart Journal. What is the definition of this hidden cardiotoxicity? But before that, I will give you a very brief introduction of drug cardiotoxicity and why is it still important and why do many groups still work on it. And there is also a LEDUC grant and a couple of MRC grants running on cardiotoxicity in Europe now. So in what is toxicity? So in every drug has an effect and has some side effects. And unfortunately, none of the drug can escape from some side effects. So we have the wanted therapeutical effect and we have unwanted effects. We are very lucky if the unwanted effect is tolerable or maybe even beneficial. But unfortunately, there are harmful toxic effects which can be pathological, for example, cardiotoxicity or any other organ toxicity, or other type of toxicity like genotoxicity and reproduction toxicity. And now we will deal with the cardiotoxicity because this is a very frequent, still unsolved issue also in the clinic and also in drug development, which is really very strange. I'm pressing different buttons, but finally I can manage to forward it. So this graph shows that the drug development course, so you have seen this in this entrepreneurship presentation. So during the preclinical phase, you are screening drugs, and of course the regulatory authorities make compulsory that the companies should screen for preclinical safety of every drug before they go to the clinic. Okay, why is that? Because we have to make sure that when at the phase 0 or the phase 1 clinical trial starts, the drug is safe. So the first man who will meet with this new compound or the new therapeutic preparation, then it should be safe and, of course, not kill the patients. However, the problem is that during the course of the clinical phases, and also sometimes during the period of the drug on the market, cardiotoxicity reveals, and this is a huge problem. So, and these cardiotoxicity reveals in patients during the clinical phases, either during the clinical trial or when the drug is on the market. And sometimes only phase 4 studies, post-marketing studies, will reveal the cardiotoxicity. And also some pharmacovigilance data. Do you know what is pharmacovigilance? Who knows what is pharmacovigilance? Yes, some know. So this is a system where when a drug is on the market, each doctor should report to the European Medical Agency, a medicines agency, if he or she thinks that there is a side effect of a drug. And it should be reported and they collect some data on it. And if the data, let's say, they are monitoring this data, and if this data shows that there is some toxicity or there is some safety issue, then the authority checks this and maybe withdraw the drug from the market. Okay? So this unexpected cardiotoxicity accounts for 60% of the drug withdrawal from the market now, and also counts for the termination of clinical trials during the clinical trials. And these are unexpected cardiotoxic effects which couldn't be revealed for the current by the current safety testing methods. And this is now the problem, and let's go on. So this cardiotoxicity of drugs is still an unresolved clinical issue. So it also contains a big clinical problem because, as you may know, there are still many of the drugs on the market which has cardiotoxic effect as a side effect. The next speaker, Pietro, will talk more about the oncology drugs and how they have some cardiotoxic effect. Here you see many of the drugs and the incidence of the cardiotoxicity as side effects. As you see here, there are some, depending on the patient group, can reach or 20% as well. But there are significant effects. These are, mostly kinase inhibitors and other anti-tumor drugs. There's also an economical problem because there are a huge number of drugs withdrawn from the market due to cardiotoxicity, and this can be very hard for the pharma companies who are producing these drugs. And in several cases, such withdrawals really gave a very hard time to pharma companies. So here I will focus later on the rofecoxib, which was withdrawn by the market due to unexpected cardiotoxicity by the manufacturer. So in this case, the manufacturer decided to withdraw it from the market. So what are the mechanisms of this cardiotoxicity? And I try to put forward that I call this cardiotoxicity what we know is some kind of a direct cardiotoxicity, which is evident. So these the mechanisms is not known. If we would know the common mechanisms of cardiotoxicity, that would be easy for the drug developers, but unfortunately we still don't know the mechanisms. These are just some literature review from one of my colleagues that showing that cardiotoxicity often involves oxidative and nitrosative modifications, and which also converge to mitochondria. So as you see, for cardioprotection, mitochondria is very important because we try to protect the myocardial mitochondria. But also it seems that most of the cardiotoxic drugs has some effect which also converge to the mitochondria, but the mechanisms are really not known. But here you see that there are several of the mitochondrial enzymes which show some dysfunction in many of the cardiotoxic effects. This is especially for the doxorubicin type compounds. So I think it was introduced first in 2005 that there are two types of cardiotoxicity. The mechanisms have been classified in two types, which has been largely criticized lately. But there are type 1 cardiotoxicity, and I still call it direct cardiotoxicity to make difference between the hidden cardiotoxicity. And this type 1 cardiotoxicity is the so-called direct, these drugs cause direct cardiomyocyte damage. And the typical drug which causes this type of cardiotoxicity is the anthracycline compounds, which the doxorubicin is one of the oldest compound which is still in use. So the mechanisms, of course, this is some assumption for the mechanisms. But as you see, the doxorubicin induces lipid peroxidation and some kind of oxidative stress, but I don't want to go into detail because then Pietro will show some more data. I don't show original data here, only some reviews. And that means that doxorubicin somehow, even from extracellular and from intracellular, they produce ROS and lipid peroxidation and also mitochondrial damage and uncoupling of NO synthase. They cause nitrosative stress and so on, but the mechanism is still not really known. So that's why, for example, now a LEDUC grant is ongoing to explore the mechanism of cardiotoxicity led by Emilio Hirsch from Torino. So the type 2 cardiotoxicity is classified to those drugs which cause cardiomyocyte dysfunction. So they don't lead to apoptosis or they don't kill the cardiac myocytes, but usually the effects is longer and less milder. According to some assumption, they are even reversible, but they are causing cardiomyocyte dysfunction. The prototype drug is trastuzumab, and this cardiomyocyte dysfunction and the trastuzumab predisposes the heart to further toxicities. For example, if the patient is treated with trastuzumab, then the anthracycline toxicity is aggravated, and it can appear even earlier. You know that the anthracycline toxicity sometimes appears 10 years later or even longer, according to some clinical data, but the next presentation will show a little bit more about it. So, the cardiotoxicity of multi-target receptor tyrosine kinase inhibitors, which are a lot of new drugs of this type. And here you see that sunitinib, for example, they are affecting the ribosomal S6 kinase, for example. They inhibit AMPK, and therefore they are inhibiting the mTOR pathway or liberating mTOR pathway. So this is also some assumption, but finally again there is a mitochondrial dysfunction and the mitochondrial damage, and some apoptosis might occur, but the mechanism seems to be very complicated, and there are not too many new data on them actually. So, but PI3 kinase no, is not here. That's the reason the PI3 kinase has been involved in some of the toxicities as well. So that's why I think that Emilio Hirsch got this LEDUC grant to further follow up on that. So this cardiotoxicity needs new classification. Definitely true. Actually, I just found this expert analysis in JACC last night, that they say that this type 1 and type 2 cardiomyopathy cardiotoxicity classifications are complete nonsense. Of course, there is a pro and con paper, an expert review on JACC, but actually I can share this view because it says that each type of drugs has different type of the mechanisms can be completely different depending on the certain drug. So for example, and therefore he proposes that if we have type 1 and type 2, this is nonsense, so we have to have type up to, I don't know, 100, depending on what drug and how it causes cardiotoxicity. Yeah, for example, you see this, the anthracycline, and then the HER2 inhibitors. These are the classical type 1 and type 2. However, the other drugs might affect the cardiac myocytes negatively by totally different mechanisms. So definitely we also think that it needs a new classification of the cardiotoxicity. So, and now, since we think that it definitely needs a new classification and a new mechanisms, we have to do something because it's a clinically unresolved problem. And basically, due to the insufficient safety testing, there are many drugs which are on the market which has cardiotoxicity and with known cardiotoxicity, and probably there are many on the market which we still don't know if they are cardiotoxic because it hasn't been revealed yet. So we have to find out some new stuff in this field, and therefore we introduced this term, the hidden cardiotoxicity. And I will tell you how we came into this assumption that and why we introduced this term hidden cardiotoxicity. We have been doing ischemia, reperfusion, and cardioprotection studies for, I don't know, 25 years. I'm that old now. So, and here you see that you have seen this figure from Rainer Schulz or Gerd Heusch, I don't know, that this is the ischemia reperfusion injury. You see that during ischemia, this is ischemia, then the short periods of ischemia is tolerable, so there's no cell death. However, if you go a little further with time, then the cell starts to die, and with reperfusion is also a reperfusion injury, okay? And you can decrease reperfusion injury by cardioprotective methods. This is what we have been studying over decades. This can be ischemic conditioning. You have heard that ischemic conditioning are effective, but depending on the comorbidities, they might not be effective. And also, by cardioprotective drugs, we can reduce the reperfusion injury, but the problem is that we still do not have any cardioprotective drug on the market. So the current drugs used to treat angina and infarction patients has not been proven to reduce infarct size. So that's why the companies are really seeking for new mechanisms for cardioprotection. However, you can aggravate ischemia reperfusion injury by cardiometabolic risk factors and sometimes by their drug treatments as well. And this is, for example, diabetes mellitus and dyslipidemia. Actually, in 1995, we were the first who shown that comorbidity, hypercholesterolemia, blocks ischemic conditioning, which opened up a new field, which is still controversial. But we also realized that in well-known cardioprotective pathways, like the N-cyclic GMP protein kinase pathway, is disrupted during hyperlipidemia. And we've also seen that some drugs, which we thought to be cardioprotective, like, for example, statins, might interfere with the effect of post-conditioning, for example. I think Rainer Schulz showed that figure. And we also seen that, for example, nitroglycerin, one of the oldest anti-angina drugs, which has been used for ischemic heart disease patients for more than 100 years, when you develop nitrate tolerance, it also blocks the conditioning effect. So we thought that some drugs has no direct cardiotoxic effect, but they just can aggravate the ischemia reperfusion injury, and they might just disrupt the endogenous inherited cardioprotective mechanisms which can be triggered by short terms of ischemia. So in other words, some drugs can block pre or post conditioning or remote conditioning, which now we know. So, for example, that propofol really blocks, for example, remote conditioning, and it seems that long-term statins, for example, can block the effect of post-conditioning, and also nitrate tolerance. If the nitrate is not given properly to the patients and nitrate tolerance develops, it can be also block preconditioning and ischemic conditioning. So it can be aggravated by drugs as well, and therefore the problem is that the routine preclinical toxicity screening methods are not able to detect these effects because it's simply done only in healthy myocardial tissue. There is a whole lot of guidelines from the European Medicines Agency how to do safety studies. You have to do it under very strict quality control, under GLP, good laboratory practice. And to do papillary muscle, arrhythmia, electrophysiological studies, but it's all required to do only in healthy tissues. And that's why we call hidden cardiotoxicity, which only reveals when the myocardial tissue is not healthy. So if you use healthy tissues with the current, according to the current guidelines of safety testing, you can never pick it up. So the definition what we put forward in this European Heart Journal paper, actually it's 2019, it just had the final pages. So we postulated that cardiotoxicity of drug that manifests only in the diseased heart, this is the definition of hidden cardiotoxicity. So either during ischemia reperfusion injury, or in the presence of its major comorbidities like hypercholesterolemia and diabetes and so on. So, and this figure summarizes this whole cardiotoxicity of drugs. So let's go over it a little bit. We have time to spend on it. So this is the drug-induced cardiotoxicity. This is a direct toxic signaling when the drug is added to the heart tissue or any other healthy preparations. It leads to death of cardiac myocytes. It leads to electric imbalance, and it can cause infarction, heart failure, arrhythmias, for example. This is typically anthracycline drugs do this, actually, or trastuzumab. And however, the hidden cardiotoxicity is those drugs can do this hidden cardiotoxicity which not directly affects the cardiac myocytes, but indirectly enhances the deleterious cardiac cell signaling, which can be coming from, for example, aging, diabetes, hyperlipidemia, and or ischemia reperfusion injury, which anyways would lead to cardiac myocyte death. So if you enhance these pathways, and this we think that COX-2 inhibitors does, and that's why they were withdrawn from the market, they can enhance this deleterious signaling, but you can see it only when there is a diseased heart, when the drug is in the drug is added to the diseased heart already, not in early preclinical safety testing using healthy heart tissue. And there is also another possible type of hidden cardiotoxicity when the endogenous survival signaling mechanisms might be disrupted due to the effect of the drugs. And this is ischemic conditioning, which initiates some survival signaling and leads to survival of cardiac myocytes, for example, during ischemic reperfusion injury. So if you block these effects, of course, you will block the ability of the heart to adapt to myocardial ischemia, and that might be also a possible way of cardiotoxicity. So this is the major theory behind it. But of course, and as I mentioned, we know already that, for example, long-term statin treatments might block the effect of post-conditioning, and we have shown also in the literature that nitrate and nitrate tolerance may block also these survival signaling pathways. And thereby, although they are on their own show some cardioprotective effect, they have also this hidden toxic effect. But how can we detect these effects? So And just again, so the clinical relevance of this hidden cardiotoxicity is that these unexpected ischemia-related cardiac adverse events are among the leading causes of discontinuation of clinical trials, and also withdrawal of drugs from the market. And why is it happening? Because there are shortcomings of the current preclinical cardiotoxicity screening methods. So drugs are not tested in myocardial ischemia reperfusion injury and under comorbidities. They are tested only in the healthy myocardial tissue during the preclinical drug development. And they can interfere with endogenous cardioprotective pathways, and this is totally not investigated so far at all. So we have to prove that if this theory is okay, so we have to find a drug which was withdrawn from the market and show that if due to clinical hidden cardiotoxicity, which reveals only in the clinic, so you have to find a drug and have to test in a preclinical model to show if it's really valid and if we could really show before the clinic that this drug might be problematic. So first we selected a couple of drugs which has been removed from the market due to cardiotoxicity and especially due to ischemia reperfusion related events. There are plenty of drugs of those drugs. And of course, probably the most famous is the rofecoxib study. Merck withdrawn this drug from the market, and because there were some fatal and non-fatal myocardial infarction due to the use of rofecoxib. This is a COX-2 inhibitor. It was used as a painkiller, especially also for young sportsmen and so on. And due to some trial, some meta-analysis and some clinical trials, the company realized that there is a safety issue here, and on its own decision withdrawn it from the market. Of course, there are still court cases, legal cases are ongoing, although this withdrawal was many years ago, maybe almost 10 years ago. I don't remember now by heart. So I was also involved as an expert in one of these legal cases of rofecoxib. So actually there were some clinical trials showing that rofecoxib increased the occurrence of myocardial infarction in patients with rheumatoid arthritis where they used this drug. And also the approved trial showed that there was an increased risk of cardiovascular events in this trial. And also a meta-analysis shown that it increased the risk of arrhythmias. So the COX inhibitors also increased risk of arrhythmias. The background, if it's ischemic or not, we don't know from these trials. So therefore, we selected rofecoxib as a test drug if we can really pick it by using a preclinical model, if it was toxic or not. So we planned to set up a hidden tox testing platform to explore the effect of rofecoxib of ischemia reperfusion-induced tissue injury and arrhythmias, and also as a second level, let's say, if rofecoxib can block ischemic conditioning, preconditioning. And the reason why we used preconditioning, because this is the conditioning type what works always in every lab, in every model, why post-conditioning and remote conditioning is not that easy. So this was a very easy experimental setup. So we treated two groups of rats for 1 month with vehicle and with rofecoxib, and afterwards we randomized the animals to ischemia reperfusion injury and preconditioning. And again, the rofecoxib group also ischemia reperfusion injury and preconditioning, and we measured the infarct size. This is a very routine protocol in the rat. We used the rofecoxib dose calculated to the rat metabolism and rat surface, so it is a human relevant dose. So what did we observe? First of all, that rofecoxib increased mortality of these rats during ischemia reperfusion injury, which is of course not healthy. And what we have seen, that ischemic preconditioning was reducing this mortality, and also ischemic preconditioning reduced the mortality increase due to rofecoxib. So that was the one major finding. So we went on and found out why they died, and there was an increase in irreversible ventricular fibrillation during the ischemia reperfusion injury in the rofecoxib treated group. So they were pro-arrhythmic during ischemia reperfusion injury. And this is, you see, this is the arrhythmia map. So when there is black, that means that the rat died. When there is the ventricular fibrillation is shown by red, and the black is when it's dead. So you can see how and when arrhythmias occurred in the vehicle and in the rofecoxib group during ischemia and reperfusion injury. Okay, so afterwards, since we picked that there is an increased incidence of ventricular fibrillation during ischemia reperfusion injury, we went to one of our collaborators to do an isolated papillary muscle where they could do simulated ischemia reperfusion injury in the papillary muscle. This method and use the conventional microelectrode to detect the action potential. Actually, this method in the healthy heart is a must to do as a safety screening, actually, but not during ischemia reperfusion. So what we have seen that in the healthy preparations, an increasing dose of rofecoxib did nothing at all. However, when there was ischemia reperfusion injury, it increased the action potential duration, which increase was aggravated significantly by the increasing doses of rofecoxib. And again, what was interesting, that ischemic conditioning protected against this rofecoxib-induced action potential prolongation. So that was the final conclusion here, that the rat died during ischemia reperfusion in the rofecoxib-treated group in a more so more rats died, and that was due to the incidence, higher incidence of ventricular fibrillation, and in the background there was an increased duration of action potentials so far. So to summarize this part, that's the case. So the action potential duration should be measured as a safety testing in healthy animals anyways, but if there are some drug effects which can prolong the action potential duration, that can be actually tolerated by the healthy heart, because they activate some potassium channels and the repolarization is secured. I'm not an electrophysiologist, so if you have some very specific question, I am sure I cannot answer. So that was done in Szeged by Professor Varro's group. And in the diseased myocardium, when there is a drug during ischemia reperfusion injury, where there is or any other diseased heart, when there is a prolongation of the action potential duration due to the other morbidity of these preparation, this accommodation, so the repolarization reserve cannot be activated, and therefore there will be arrhythmias. So this is at least one point what we think is in the background. But let's go a little bit further and let's see what happened with the infarct size of these rofecoxib treated animals. And this is interesting. that so we have seen that rofecoxib killed the rats with ischemia reperfusion injury because of arrhythmias, but in the meantime it reduced infarct size, and of course we thought, and also the preconditioning effect was not touched basically, so preconditioning was still working in the infarct size. However, this could be an artifact. Why it could be an artifact? Yes, it's a question to the audience. Yes. Yes, because here in the infarct size, the rats should survive. Okay, so in the rofecoxib group, there are a lot more animals were dead, so we excluded those rats who were dead, and therefore it could be an artifact that since we are measuring infarct size only on the survivor animals, therefore we had to check if this rofecoxib effect is really true and it acts directly on the myocardial cells, because otherwise we cannot exclude this artifact, what you pointed out very correctly. So therefore we went to cardiac myocytes experiments, and we subjected the myocytes to simulated ischemia reperfusion injury and tested different doses of concentrations of rofecoxib. And definitely what we have seen is that rofecoxib showed a bell-shaped cardiac myocyte protective effect during ischemia reperfusion injury. So the picture is pretty complicated. So by now what we have shown is that rofecoxib increased acute mortality due to the irreversible ventricular fibrillation and increased the action potential via probably increasing the action potential duration. And by this, actually, we could show that rofecoxib has a hidden cardiotoxic effect, because this pro-arrhythmic effect was only evident when we exposed the ischemic heart to rofecoxib, and it was not seen, not any effect was seen in the healthy myocardium. Another interesting phenomenon, that ischemic preconditioning protected against this rofecoxib-induced hidden cardiotoxic effect, but on the other hand, rofecoxib was able to directly protect against ischemia reperfusion injury, so it has some mixed effect so far. But nevertheless, based on these results, we think that this is the first demonstration that preclinical cardiac safety testing of drug in the presence of ischemia reperfusion may reveal hidden cardiotoxicity of the drugs, and we are planning a lot more of these experiments in combination of ischemia reperfusion injury and also some other comorbidities as well, with like hypercholesterolemia and diabetes, and see if the test compound rofecoxib can be shown as a hidden cardiotoxic drug. And of course, we postulate in the future, when we will have enough data, that definitely the preclinical safety testing of the drugs should be done not only in healthy myocardial tissue, but also in the diseased myocardial tissue, which includes ischemia reperfusion injury and several comorbidities. Actually, I think it's very trivial, but so far is not required. So that's why, also science needs to be turned into business, as we heard, that we are planning to develop a hidden tox testing platform, because we think that with the rapid testing for this hidden cardiotoxicity, what can be the benefit for the patients. So let's see what are the benefits for the patient. As in the beginning, I have shown that hidden cardiotoxicity revealed in patients during clinical trials and in post-marketing studies. Of course, this is not a good sign because it should be predicted much earlier in the preclinical phase. So if we would develop some testing platforms and some standardized testing platforms in the preclinical phase, then we could increase the safety of the drugs because we could save these patients here. And of course, we save the development time because if we, for example, let's get the example of rofecoxib. If rofecoxib would have been tested under ischemia reperfusion in the preclinical testing, for sure that this drug would be not indicated for ischemic heart disease patients, for sure. And actually there are other COX-2 inhibitors on the market, and there are some COX inhibitors which has very similar effects than rofecoxib still on the market, but in their marketing authorization already there is a risk for ischemia reperfusion is written now. So by this development we could either block the development here, or at least we could predict a much safer use of these drugs. And of course that saves a lot of development time as well. And of course by this testing platform we could increase the success rate of development, because just imagine when you develop a drug, spend money until this point, and then suddenly cardiotoxicity appears and the clinical trial should be immediately terminated. Or the worst cases when you enter into the market and after 1 year of sales, the European Medicines Agency says that that's okay, so you have to withdraw it immediately from the market because of some unexpected cardiotoxicity. And of course, there's a lot of saving of the development of the costs if we would do such kind of testing. So now we are doing a couple of studies. This is the previous study I've shown is still unpublished. We are just submitting the paper soon. We don't know what is the mechanism of the rofecoxib-induced cardiotoxicity. We have done a lot of omics study to find out the mechanism because we have no clue. And the other thing what we do, and actually my colleague Mátyás Pétervár has a poster on it, that by using the European Medicines Agency Pharmacovigilance database, you know what I told, what is pharmacovigilance? This is a compulsory data gathering of the European Medicines Agency. Every doctor has to report any suspected side effect of any drugs on the market. So by using this pharmacovigilance database of the European Medicines Agency and by using a network analysis method, we are trying to find some connections between some compounds on the market which can be related to acute myocardial infarction, and maybe by using these databases you can also find out the hidden cardiotoxicity of some drugs which are already on the market, but the pharmacovigilance databases has not yet picked up that it's significant. So there is it's a very difficult data analyzing method to show connections, but some good mathematicians and our new bioinformatic group does such things. So, and by this I just thank you. This is my group in Budapest, and we are looking for PhD students because the government increased the number of state-funded PhD students by 50% from September, so it will be difficult to fill up. So thank you very much for your
Thank you, Peter. Are there questions from the audience? Yes.
So very nice lecture. Thank you for that. I have a question about the celecoxib. It's in Poland. We just saw it in the pharmacy. Can you tell me about it more? Because now my feeling is mixed about.
Yeah. I can say only my opinion, because I cannot override the European Medicines Agency, marketing authorization policy. That I think that there are a couple of drugs on the market which has a very similar side effect, let's say, pattern than rofecoxib, but is still on the market. Now we know better. Now we know more, and I think in the celecoxib, summary of product characteristics, I think that there is a risk of increased infarction, and it's also in the caution point. There is written that for ischemic heart disease patient it can be used only with caution. That's as far as I remember, but I don't know exactly. But I think that's the case. But I know, but yeah, but the diclofenac has very similar actual effect on COX-1 and COX-2, which is, of course, our drug is still on the market actually. But now we know the caution. So I again repeat that it was not withdrawn from the market by the European Medicines Agency, but by Merck.
Hi, great lecture. I wanted to ask, so after, during your presentation, in the ending part, you seem to show that it was ultimately the electrophysiology which you used to find out that it has a prolongation of the action potential duration and it causing arrhythmias. Also, there are agencies around the world, in America and in Japan, for example, the JICSA organization, which they are trying to standardize their way of testing cardiotoxicity. So, in your opinion, is the world actually moving towards using this kind of, for example, the MEA devices or Thomas Hagen's EHTs for kind of some standardized way of testing the drugs, or like do you still say that animal models like the rat studies will still be used, or how is it going to be?
Yeah, look, we believe that the world should move in this direction, and definitely if we will gather enough data, I hope that in the guidelines it will be included that safety testing should be done in diseased tissues, including electrophysiology and including, for example, ischemia, reperfusion-induced cell death, and so on. But we need more data. As you saw, this was very mixed data, and with rofecoxib we could see the pro-arrhythmic effect only during ischemia reperfusion injury, but rofecoxib itself showed some cardioprotective actions as well. So. And also there might be many drugs which just block the preconditioning or the conditioning phenomenon.
Thank you very much. Excellent talk. I would like to ask you something. If we manage at least to put this model to test the drugs for cardiotoxicity, wouldn't it be safer to have a threshold? I mean, let's say an algorithm, put all the data together from electrophysiology, Infarct size, maybe some more parameters, and learn the model what drug could be cardiotoxic or not. I don't know what your opinion is.
Yeah, of course, actually this more or less exists, but because there are a lot of data in the literature, but the problem is that the preclinical testing is entirely an internal issue of the developer company, and sometimes they don't publish anything. So what is public is, but that's gonna be changed actually as well. So there is an initiative of the FDA and the European Medicines Agency to make available all clinical trial data as well. And also the pharmacovigilance database. This is very important, but in certain countries the doctors are totally neglecting it. So, for example, unfortunately, my country is a good example that, for example, compared to Germany, a paracetamol side effect is reported at least 10 times less than in Germany. So maybe the Hungarians tolerate it more, but I don't think so.
Yeah, I have a question. I mean, with regard to the mechanism, and it also relates to the comments of some of you. I mean, one of the problems I think with cardiac toxicity is its wide range of mechanisms, very, very different mechanisms. And originally, cardiac toxicity has been considered to be a direct effect on myocytes. But I mean, you picked the example of rofecoxib, and from my understanding of the whole story, that has been a predicted cardiovascular toxicity. Garrett Fitzgerald, for example, already published 20 years ago that if you change the ratio of COX-1 and COX-2, and you block COX-2, which is protective in terms of prostacyclin production, and you do not block COX-1, which is responsible for thromboxane, then you change the ratio and you induce infarction. So what is your evidence? I wanted to ask you, what's your evidence that this is a direct effect on the heart versus, let's say, the expected effect on platelets and endothelial cells? Or even more specific, your rat study, has it been done in situ or in perfused heart?
No, it was in situ, and of course
so
with anticoagulation, so it was heparinized animals. So this effect, I don't think it might be a problem. And the other thing, I know that it was suggested that it's a platelet aggregation issue, but it's not definitely that, because there are some studies showing that there were pro-arrhythmic effects of there was one meta-analysis I showed.
But this can be easily explained by ischemia.
It can be explained as well, but there is, as we showed, there is also prolongation of the action potential in the pure preparation of the papillary muscle. So
what's the expression of COX-2 in myocytes?
Yeah, we haven't checked here the expressions of COX-1 and COX-2 in these preparations. Of course, this is, since we don't know the mechanisms, we have already transcriptomic data between the control and the rofecoxib animals, but just to tell, it's still confidential, but we didn't see too much expression difference. So the mechanism should be further going on, maybe on proteomics and phosphoproteomics, or I don't know. So mechanism is very, very unclear.
My question was also in line with the one with Thomas, with COX-2 is constitutively expressed in the myocardium and the vessels. So perhaps this highlights also that clinical trials should be well predefined before starting. Because this came out when I was doing my PhD with Lina Vanimon, and I remember us talking together in a dinner, and we said, it's not going to work because they are blocking COX-2. How can they come and just include everybody within this trial? This was like late 90s. So the thing is that probably this also highlights that your studies of this kind of approach may also help a lot. They have to be mandatory.
Plan better clinical trials. Yes, that's what I suggest. So more clinical trials in the presence of comorbidities will help to plan more successful clinical trials on the one hand. On the other hand, you save patients because you exclude those patients where the toxicity would be higher. So.
Peter, great. Now, how patients usually take even 10 medications per day? What's the role of multitherapy? You know, it may be that one drug doesn't have a cardiotoxic effect, but in combination with another drug, it may trigger.
Yeah, that's a good question. This is typically a mathematical question, so that's why we build up now this pharmacovigilance bioinformatic group to dig into this pharmacovigilance databases and with the mathematical network analysis find out new interactions and new side effects.
So we move to the next speaker. Thank you. Thank you.